Good evening, ladies and gentlemen, and welcome to a very special edition of Battling and Beating Cancer here on CanTV 21 in Chicago, and we're also streaming live worldwide at www.chicagobloodcancer.org and cantv.org. And I have to tell you that as one of the co-hosts of the program, this is the best show that we are ever going to do because of our terrific guests. I'm Scott Seaman, and along with Charlene McMahon, who will be replacing me on your screen in a couple minutes, we are co-hosts and co-founders of Chicago Blood Cancer Foundation, and we welcome you to our show. And when I say we have a very special guest, we really have a legend. We talk on this program all the time about how cancer research is the key to all of our long-term survival, and how blood cancer research really is a superhighway to curing cancer, because virtually every major breakthrough and treatment for other cancers, as well as the blood cancers and other diseases, have come from lymphoma and leukemia research. And we also say that the superhighway to curing cancer runs right here through Chicago. And the reason is because Chicago has contributed so much to research. And when we talk about research and cures and helping patients, we are really talking about Dr. Janet Rowley from the University of Chicago. And Dr. Rowley really personifies the marvels of research. And she is one of the most humble and nice and kind people you're ever going to meet. And because she's so humble, and because if I spent time telling you about all of her achievements, we'd be out of time. And you're going to want to hear what she has to say about lymphoma, leukemia research, and all of the magnificent things that she's done, and tie it together with how what she's done in the laboratory has translated into magnificent treatments for people. And Dr. Gregory was on a couple weeks ago, and you'll recall that we were talking about CML, and how years ago people that got diagnosed with CML, it was a virtual death sentence, a very short life expectancy. Well, now people with CML are living well, and they're taking medication in pill forms, Gleevec, and now there's even a couple other types of these kinase inhibitors. And as a result, they're living 15, 20 years and still going strong. And all of that stems from the magnificent work that Dr. Rowley has done. She's been called everything from the matriarch of genetic research for cancer, because she really put genetic research on the map, and she continues to do so many marvelous things. And as much as her research has done and led to cures and better treatments and other discoveries by other scientists, because it all builds on everything else, one of her major contributions, I think, Doctor, is all of the young researchers and now not so young researchers who you have influenced and who you've worked with and encouraged over the years. So I have to tell you, as a cancer survivor, I'm so grateful for all that you've done and continue to do. But I'm not the boss. I have a boss. Her name is Charlene, and she outvoted me. We were fighting to interview you. She won, so I'm going to turn things over to Charlene, and we'll talk to you about some of your great work over these years.
Okay. Dr. Rowley, it is just great to be with you. Tell us about how you became interested in cancer research.
That was, if you will, by accident, because I started to do research in England, which I wanted to continue on. And I had graduated from the University of Chicago and knew a number of professors, and went back to them and asked, could I have a laboratory? Could I keep on doing my research? I wasn't interested in doing somebody else's. And by the way, would they pay me? Mm-hmm. And Dr. Leon Jacobson, who became chair of medicine and then dean of the medical school at the university, agreed to all of my conditions, including paying me. And he was a hematologist, and so I worked in space that he controlled. And it was known that chromosome abnormalities were important in one kind of leukemia, as Scott has already mentioned, CML. And so when those patients came to see Dr. Jacobson for treatment, he would ask me to do chromosome studies on them, which I was more than happy to do. And that's how I began.
Wow, what a story. Start the beginning of terrific. In 1970s, you identified the translocation of chromosomes that resulted in AML, chromosomes 8 and 21, CML, the Philadelphia chromosomes, and chromosomes 9, and later on some lymphomas. Tell us what translocation of chromosomes and what your discovery was with the translocations.
Well, translocations can involve different chromosomes, and those that are important in cancer are relatively specific to particular chromosomes, so they're not just random. And what a translocation is the break in a chromosome. And if you think of a big X or a big V, that's what chromosomes look like in a human cell. And a piece of one of the parts of the chromosome can get broken and switched with a broken piece of another chromosome, and that's a translocation. So it's really a translocation of a piece of DNA from one chromosome to another.
And you did a lot of, I know you did a lot of this work starting like on your dining room table, if I'm correct, from what I've read. Yes, that's where you started. One of the significant aspects of your discoveries is that you show that cancer was a genetic disease, and I imagine that was a bit controversial at the time.
Well, that's very true, because back in the 1970s and the 60s preceding that, it was assumed that the chromosome abnormalities that people had seen in cancer cells, which differ greatly from one cell to another, that these chromosomes were a consequence of the cancer process rather than a cause. So to turn around and say in the 70s, following the discovery of translocations, that these specific changes in different leukemias and lymphomas, and now we know in solid tumors as well, that they were integrally related to the cause of the disease, that was not part of the current paradigm at that time.
Okay. Your work led to better diagnostic protocols and treatments of patients. So, for example, as Scott was saying earlier, people diagnosed years ago with CML did not have long to live. And but today, because of Gleevec and now other pills, people are living well and for a long time. Could you explain briefly how we got from your discovery of translocation of chromosomes to life-saving treatments?
Well, one of the questions when one discovered the translocations was, though the same chromosomes and apparently parts of chromosomes were involved in different patients with the same kind of leukemia, were the breakpoints in different patients the same, and what was at the breakpoints and why was it important in the disease? And many people discovered genes, and many of them genes that were carried by viruses that cause cancer and leukemia and lymphoma. And what we could see was that the genes that were important in animal cancers that viruses had picked up were also the genes that were very important in patients, and that the breakpoints weren't random up and down the length of the chromosome, but they involved not only particular genes, but very special spots in those genes. And so you'd swap ends between a chromosome carrying a gene that was active for cell growth, and particularly cell growth in bone marrow cells leading to leukemia, but it would swap pieces with a gene that made it a signal for growth all the time. So instead of this gene being under control that was very often a brake and stopped growth, it would become a gene that was signaling with the gas pedal down, signaling growth all the time. And what Gleevec does in the case of a translocation in CML is to inhibit this growth, constant growth signal, such that a patient's cells can respond to the normal signal and the disease goes into remission. And that's what's been so important about Gleevec and the second and third generation tyrosine kinase inhibitors that Scott and you have both referred to.
Wow. The immunotherapy of today, like monoclonal antibodies and kinase inhibitors, have been very effective and can less side effects because they do not harm healthy cells. Is that correct?
That's true. And that's really been important in Gleevec, that patients have relatively minimal side effects and can tolerate the pills. And some patients appear to have to take the pills for many, many years. So it's important not to have side effects in that.
Your lab is analyzing the genetic consequences of recurring chromosome abnormalities in leukemia and lymphoma cells. Tell us what you are working on, like what your research techniques are you are using today, like SAJ, analysis of serial analysis of gene expression in microRNA?
Well, that's true. Now, the SAGE techniques that we used several years ago we're not using anymore because science has moved on. But we are working now on microRNAs. And so we know the genes that are abnormal in cancer cells and leukemia cells. We know many of them. And the Genome Project that is so important for all of us in cancer is looking to sequence the DNA from many different cancers, trying to see what changes are similar in similar cancers, what changes are different in different subtypes, and what that could mean in terms of how those genes are expressed in proteins and what regulates that gene expression. And so microRNAs are one form of regulation of gene expression. And it was really only discovered that they were important in cancer less than 10 years ago. And our hope is that the microRNAs, which are very small RNAs regulating the expression of genes, that maybe they might be a way to target cancer so that it might be, and they might be an easier target than some of the genes themselves. This is still ongoing research and is actively contributed to in the laboratory by many people. And I want to pick up on something that Scott said, that I'm a very old person, but what keeps me somewhat younger is having young people to work with who ask you questions and who push you to think of things that you wouldn't think of otherwise. And it's just such a joy to have them around. And then when they go on and do something and are successful, you just have such a marvelous feeling. So
well, they are very lucky to have you too. Very, very lucky. So it works both ways.
Well,
yeah. Now, because you started your work, of course, many years ago in the 1970s, how would you say that the work's changed over the years and have the techniques progressed since the 1970s? I'm sure you see a really big change.
Well, there's been an enormous change because when I was first starting, our way of picking up genetic changes in cancer was by looking at chromosomes. And a big chromosome can have 100 million base pairs or more in it. I mean, that's just a huge amount of DNA. And we could only pick up small changes in chromosomes, maybe 5 million base pairs or something like that. Again, that's hundreds of genes, and that was as close as we could get to genetic changes. Now, people sequence the DNA, you know, down to the nucleotide pair in the DNA exactly what the change is, whether it's a deletion or whether it's an extra copy or whether it's switched places with something else, at a level of precision that we couldn't begin to approach several decades ago.
Wow. You're just overwhelming me here with all this information. But what areas you think, because you focus on future cancer research, and what are some of the advances you hope we will see in the next few years? Like, nothing makes me sadder, and Scott will tell you I'm a very big pediatric cancer advocate, and nothing makes me sadder than seeing a child sick or dying from cancer. So I am most grateful for the impact that you have made in your work with children with leukemia. Can you talk a little bit about that? I mean, just, I deal a lot with children and stuff, and just recently this week there was 2 children that I keep contact with that passed away from this disease.
Wow.
So can you just put a little, spin on that so we can kind of get an idea what you do with children?
Well, in terms of childhood leukemia, there have been great advances through the collaboration with St. Jude Children's Hospital in Memphis and the Washington University St. Louis Genome Sequencing Center to look specifically at the genetic changes in childhood cancer. And they've been able to identify different subsets based on the fact that certain of the cancers have the same genetic change and also often the same morphology or immunologic markers. And this then leads to this question of what are the consequences in a cell of these genetic changes? How is the protein different? Or is the protein overexpressed or underexpressed? And this comes back to what I was saying about the regulation of the expression of these genes. But with all that information, you can now, doing experiments in animals and elsewhere, figure out what are the best treatments, what are the best inhibitors, or what can you do to, if you will, jazz up an alternative pathway that can bypass the deficiency. And it's a very complicated problem, but we're making a lot of progress.
Mm-hmm. And you talked about, because you work with young researchers, and that what would you give as far as advice to any young researchers that are interested in genetic and cancer research?
Well, science is hard, and it's not hard necessarily only because of the subject matter, but because you have a lot of disappointments along the way. You'll think of a good experiment, only it turns out not to pan out the way you had hoped or think. So you have to have a lot of personal resources to be able to survive the down periods, as well, of course, as to be rejuvenated by the successes that you have. And so I think people have to choose something that they're really passionate about. And if you don't really believe in something and go home from the lab and keep thinking about that problem and how should you attack it and what does it mean, if you don't have that kind of dedication, then I think you shouldn't go into science, because it is a tough taskmaster. And my husband used to say that, in fact, he thought scientists were like a monastic order. You really have to not only focus, but your time and attention are really dedicated to that particular problem. And if you don't have that kind of dedication, you may last through the good periods, but are you going to survive the bad ones?
Yeah, it's the ups and downs.
Yes.
Chicago Blood Cancer Foundation, we have these little buttons that say, I think Scott had one on when he was over here before, say research saves lives. But I think we should make some that says Dr. Rowley saves lives,
Well, but that makes it too personal, I think.
Well, I have to say, though, if we could clone you, maybe about 10 of you, we could probably conquer the world here. So what I wanted to talk about, too, because Chicago Blood Cancer Foundation focuses a lot on private fundraising, because that's so important, like we've talked about, too. Private fundraising is very important, and those of us that are not as talented as some others like you, through raising and donating money for blood cancer research. Can you explain the importance of private funding to, researchers like yourself?
Well, that's a marvelous question. And it's sort of like saying to a starving person, here's a dish of ice cream, or something of that sort. Science right now is very much threatened by the present fiscal crisis and the sequestration threat that's likely to take place within the next week or so. And at risk are funds from NIH. So we have scientists working all over, not only our own country, but around the world, but mainly in terms of NIH funding our own country, that are dependent on government funding for basic research, which is what much of cancer and leukemia research is. There's so much we don't understand, and in order to be able to have the thousands of scientists working on these problems, we really do need a great deal of money, and the techniques we're using in DNA sequencing, for example, are all expensive. And in my day, reagents were very much cheaper than they are now. So I look on private philanthropists as really venture capitalists, and you're hoping that some young person that you fund will find an answer like Gleevec that will transform the treatment of a disease for a whole community of cancer sufferers. And we have no idea now how many Gleevecs will go undiscovered as the federal government reduces funding for research, and that deficit in funding from the public sector has got to be, or I would hope, would be filled by public philanthropy. And I think one of the important things of foundations like yours is that nobody has, or few people have the resources to give a lot of money. So, as with Obama's first campaign, if you have a lot of people giving a little bit of money, you can have vast amounts of money for research. And that's what the goal has to be.
You are incredibly humble and have received every conceivable award and recognition all the way up to the Presidential Medal of Freedom. And what do you see as your legacy in terms of cancer research?
Well, I do think that finding recurring chromosome changes, which do represent genetic changes in cancer, was probably the most important contribution, because it did focus people's attention on the genetic changes in cancer. And all of the work that we've done subsequently has confirmed that that's so. Right now, of course, with DNA genomic sequencing of cancers.
Well, Doctor, it's about time to wrap up here, and I wanted to thank you so much. You are truly a national treasure. And I wanted to thank my husband. Of course, I always have to thank my husband. You know how that is. Yes. But I do want to say one personal note, and I think this is really amazing. This woman, she actually rides a bike to work most of the time. And I am totally amazed by that. We were talking about that before, and her husband does, and her son. And I think that's a big accomplishment right there, let me tell you. But I want to say thank you, and we wanted to say thank you to Christy and Dorothy and our CAN TV crew, and we will be seeing you next week with the episode of Battling and Beating Cancer. And good night to everybody.